New techniques to modify biology, primarily at the level of DNA, with useful purpose, right? So that I'm going to set the whole thing up by saying that, and feel free to knock that down when you have your turn. So we have, of course, been modifying biology for, well, more than 10,000 years at a genetic level with farming. But in the 1970s, there was a fundamental shift in how we did that with the advent of genetic engineering, genetic modification, with the ability to transfer genes from one organism to another. And then arguably, there's been another shift, a major shift in genetic engineering and our ability to manipulate biology in biotechnology this century. And I think arguably the biggest shift was a conceptual one, that it was the application of engineering principles and mathematical engineering principles into the modification of biology. And that, I think, is the nearest we can get to talking about a definition of synthetic biology. So, I mean, that's fundamentally setting up what it is that we're doing tonight. After each presentation, there's going to be one simple, possibly simplistic question embedded in the things that that each of the speakers will be talking about. And what I'm going to do is I'm going to reiterate that question, or state that question after they've finished, and we're going to do a show of hands vote. So they're going to be largely binary questions. So we'll do a show of hands vote, and then the next speaker will make their case, and we'll do the same times 4. So, Robert, perhaps you would like to start with food.
Thank you very much. I'm missing one slide there, I think. Okay, good evening, everybody. My name's Rob Edwards. I'm the Head of Agriculture, Food and Rural Development at Newcastle University. My own area of research expertise is actually crop protection. At the moment, we're battling with herbicide resistance in blackgrass. But one of my interests, both former and currently, are new technologies that we can bring into food security. And that's what I really want to talk about this evening. It's all around how we see food and synthetic biology and those words sitting together, and what conjures up images with those words. And this is one of them, the caravan. It's something, I think, which sets a scene in some people's minds with respect to what they see as meaning in synthetic biology and food. We know for a fact that when there have been dialogues with the public, they're particularly sensitive to issues around diet and food and synthetic biology. But that's one scenario which we could see synthetic biology impacting on food. You know, we can produce it in cultured vessels. We make stem cells, for example, to make burgers. We can garnish them with genetically modified salad, and then we can make a nice polysaccharide bun extruded from a reactor. All of these things can be done, and they can possibly be done very sustainably, but are they desirable? I certainly don't think so at the moment. Instead, we could think about synthetic biology as being a raft of different approaches that we can bring to bear to one of the most pressing demands we have in terms of a global challenge to us as mankind, our response to the food production challenge over the next 30 years. And so here we're really talking about utilizing the very best quality science and engineering approaches that we have to come up with some rational solutions to what is an extraordinarily large challenge for us to take on in the next 30 years. And this next slide gives us some kind of idea of the scale of that challenge. Every day there are 200,000 more people on the earth born. They've got to be fed. That means in 2030 we're going to have an extra 2 billion people to feed. The land that we have available to us is pretty much fixed. We can put a bit more into agricultural production. The environmental costs of that would be extremely undesirable. We're already using 40% of the land mass of the earth for agricultural purposes. So we have a real major challenge on our hands here. How do we feed all these extra people in such short order? And the slide at the bottom there illustrates in sort of diagrammatic form what that means. It means a hectare of land that was producing enough food for 2 people in 1960 is going to have to produce enough food for 5 people in 2025. Now, I'd like to be able to tell you that, you know, as agricultural scientists, we're all ready for the challenge, and we've got all of the answers in our back pocket. The simple upshot is we do not. You know, this whole area has been underfunded now for a quarter of a century, and it's only the last 3 to 4 years we've really seen significant funding going into agricultural research again, both in this country and probably more widely across the developed world. So we have an enormous challenge on our hands, and importantly, the technologies that we're currently using are being pretty much maxed out in terms of what they can deliver for us. In advanced agricultural economies, as we have in the UK, we're now seeing the plateauing of many of the yields of our major arable crops. This particular graph is winter wheat, and it's shown that since 1995, despite the best ministrations of farmers, we have a very advanced farming community here in the UK. We're using lots of pesticides, lots of nutrients. We are hitting a plateau of yield productivity, which we don't entirely understand what this yield plateau is about, but we know it's a glass ceiling that's extremely difficult to push through. That's not good news if you've got to face up to all of those issues that I was talking about earlier in terms of increasing food production. So the point is, we're not in great shape now, but we do know how to do this. In 1947, Norman Borlaug and others took on the challenge of producing more food for the earth in a concerted way, which brought to bear all of the best technologies that we had in engineering, in chemistry, and increased global production of arable crops in many instances by up to 10% per annum. And that improvement ran right the way up into the 1980s. It secured food security for the planet pretty much for half a century. And now we've run to the end of that cycle. We now need effectively a second green revolution. Now, the core technologies that we needed to deliver that green revolution are based around mechanization, chemical inputs, fertilizers, crop protection, and of course, genetics. And I think synthetic biology can make small but significant improvements in each of those, which will help us take us forward to match those challenges over the next 10 years. So I'd like you to think about synthetic biology at this point in time of a kind of human development in much the same way that people didn't think about synthetic chemistry at the advent of the 20th century, because many of those advances in the first green revolution were underpinned by enormous advances in synthetic chemistry. It gave us agrochemicals, it gave us improved fertilizers, it produced much more efficient fuels to drive mechanization and agricultural systems. And importantly, in the food sector, it added enormously to the value and pleasure of eating foods, as well as improving the shelf life of many of our foodstuffs. This innovation was introduced in a generation. So at the beginning of the 20th century, there was very little chemistry going into agriculture. By the 1950s and 60s, it was flooded with it. It revolutionized the way in which we cultivate our crops and produce our food. So I'm thinking here, this is a model that we can think about how we might be able to utilize new technologies like synthetic biology in much the same way we used synthetic chemistry 100 years ago. So where do I see the inputs coming in? In the first 10 years, they're going to be incremental. I think we're going to see a move away from many of our dependencies on chemistry to biological intervention in terms of crop protection, for example. I can see us also needing to use bioenergy produced through synthetic biology-based processes to drive mechanization, which is a very highly energy-dependent process in agriculture. Nutrient recycling, currently we're just wasting a lot of the nutrients that we put on the ground. I can see there that biological reprocessing on an industrial scale would be an enormous benefit to agricultural sustainability. Diagnostic devices, and importantly, I can see already developments where we can see synthetic biology improving the shelf life of food, helping us to reduce food waste. So small incremental increases in how we apply this new technology to help us make our food supply more sustainable and resilient. I think after 10 years, we don't have a crystal ball to see where everything is going to go. But I think, first of all, synthetic biology is going to tell us a lot more about engineering desirable traits into plants. We need to fundamentally change the crops and the animals that we eat to better reflect the lifestyles that we live today. We eat too many calories, but we have too few nutrients with it. We need to engineer our foods, perhaps in the first instance utilizing conventional breeding. But as the challenges grow greater, perhaps we start to need to think about using synthetic biology to accelerate the rate in which we can improve our foodstuffs. And we can also improve important input traits into agriculture, making it more sustainable. So the question I'm going to phrase broadly, and I've got a more specific one that will be asked in a moment, is around this idea. We live on the edge of a trilemma, this depletion of resources, increasing population, and an unstable climate. And I would put it to you that we have such major challenges in this whole sector that we would be very ill-advised not to turn to synthetic biology amongst other important technologies to help us through the next few years. Thank you.
Thank you, Robert. So, wait. So the question, so if we just raise the lights for just a second so we can just, I mean, this is a show of hands, so it's not particularly scientific, but the question that Robert's posing that I'm going to ask you now, and then we're going to repeat at the end, is this. Would you eat animal or plant-derived food with scientifically evidenced unique health benefits, which have been prepared by clearly labeled processes that involve synthetic biology? Okay, so do I need to repeat that? Yes, we do need to repeat that. Would you eat animal or plant-derived food with clearly labeled processes that involve synthetic biology if they have unique health benefits associated with them? So I want you to raise your hands if you would. Well, you can leave. If anyone changes their mind after this, then clearly you've failed. Who wouldn't? Right, so the vast majority of you would. Maybe the question was posed wrong. Maybe we should ask a more controversial question. No, okay, right. So that's useful to know. So again, thank you to Robert. Now, so that's covering food. So the second theme is going to be waste. Louise, will you take the mic?
Thank you. Yes, so I'm Louise Horsfall from the University of Edinburgh, and while I live in one of the most beautiful cities in the world, we came fourth recently, I spend my holidays exploring some other parts of Scotland, and this year I went to Mull. And Mull is a very beautiful part of the world. It is an island that is over half the size of Greater London, but it has 3,000 times fewer people living on it. So with all that space, you wouldn't expect them to quite have so much of a litter problem. But the coast of Mull, of course, accumulates plastic waste, as does any other coastline in the world. So it's a real problem, and it's a problem that's here to stay. Because even if we introduce a 5p plastic bag tax, that waste is going to be here for hundreds of years. And the reason that waste is just so recalcitrant is because it's man-made waste. So there's no natural system that's been able to evolve to break it down, to use it as its feedstock. So synthetic biology provides us with the tools and the techniques to design and engineer microorganisms so that we could actually degrade the plastics which are present that we've put into our oceans. So my question is, should we? Basically, should we? So I also wanted to highlight that using biotechnology to manage our waste is not a new process. It's something that we're all actually doing. We are diligently collecting our kitchen waste, and it's going in anaerobic digesters. And from that, we are producing fuel and energy that's being used right now. So synthetic biology offers ways to do this more efficiently and perhaps more robustly as well, because just as this kind of uses this whole blend of bacteria that you might find similar to a cow's stomach, that is a really balanced, refined balance that exists, whereas synthetic biology can offer some kind of robustness to that process. So in case something in the waste would reduce that efficiency, synthetic biology can restore that balance. And in that respect, it doesn't actually require any genetic modification. That's synthetic ecology. So the research that I do is a little bit to do with waste. That waste provides the energy for bacteria to reclaim metals. And one of the examples that I'm working on involves whiskey. And who doesn't want more whiskey in their life, right? So, yes, basically, whiskey is double distilled. And while the whiskey goes up the still as it evaporates, what's left in the bottom is highly nutritious. It's dead yeast and unfermented sugars, but it has dissolved copper in it. So that dissolved copper has to be removed before that waste, that whiskey byproduct, can be used as animal feed and fertilizer. So what I'm doing in my lab is engineering bacteria to produce copper nanoparticles from that dissolved copper more efficiently, so that we can actually use these nanoparticles in other industries, for other uses, for other purposes, completely separate to synthetic biology. Another example of the research that I'm doing as part of a much larger collaborative project is to try and incentivize the decontamination of land. Land that's through decades, centuries of industrial use has been contaminated with metals. If we can use the natural process that plants have of accumulating those metals, we can then take that plant waste and process it, reclaim the metals to help incentivize the whole process, the land decontamination process, because it's a very long process when it involves plants. It would be quicker if we could engineer the plants, but we'll skip over that bit. And basically, the reason why this is so important, why our waste management needs to change, is because we currently live in a linear economy. And this planet has a finite amount of resources. The UK in 2012 was estimated by the Green Alliance to be 81% linear. So we're not recycling enough, we're not reusing enough, we're not remanufacturing enough. So synthetic biology offers a whole new take on this. Not only could we use living systems to help recycle our biological nutrients, we could actually use synthetic biology to engineer living systems that also help us with the waste problems of non-biological nutrients. Thanks.
Thank you, Louise. So let's do the poll again. Thank you, Louise. Let's do the poll again. So pop the lights up now. She stated the question. I'm just going to reiterate it. Should we engineer microorganisms for use in our oceans to degrade the man-made plastic waste found there? Now, I did stress in our oceans. So we're talking about releasing synthetic biological organisms into the wild. So if we think, yes, we should engineer microorganisms for use in our oceans to degrade the man-made plastics, please raise your hands. See, this is the problem with doing things at the Royal Institution, really, isn't it?
That was really controversial.
So can you just do that again, please? So I'm, and can we? And who thinks we shouldn't? Well, I'd say that was roughly 50-50.
Can I give a reason why?
You can later, madam. So as soon as all four have finished, I'll be opening it up to audience questions, and the majority of the rest of the evening will be devoted to interactions between us. So yes, I think we've got roving mics for later on, yes. Anyway, thank you, Louise, again. So that's food, waste, Paul Freemont, health.
Yes, good evening, everyone. My name's Paul Freemont. I'm the co-founder of the Imperial College Synthetic Biology Hub, which is the Synthetic Biology Research Centre, and it's also the National UK Innovation and Knowledge Centre for the translation of synthetic biology into industry. So I'm going to pose a lot of what-ifs and just sort of open up this debate about synthetic biology. So what if you could take a yogurt drink that had engineered bacteria in it, and then you went to the toilet, obviously one does, and then one looked at the colours of one poo, and that the bacteria had reacted with your stomach, changed color, and produced different colored poos. This is sort of what I would call my cheap personalized disease monitoring from the inside out. And that was a really fantastic project, by the way. It's just been published. Anyway, what about this one? So engineered bacteria. So if we were to systematically engineer bacteria, E. coli, for example, and there are really safe versions of E. coli, and you could eat them or whatever, and then they could sort of find their way into your colon and seek out colorectal cancer, and then somehow interact with those cells and actually target them directly to remove that cancer. What if you could design a bacteriophage? So bacteriophages are natural predators of bacteria. But let's engineer one. Let's really engineer it systematically so that it can attack pathogenic bacteria. It can actually go and look and seek out those bacteria which are harmful to us, attack them, do what phages do best, destroy that bacteria, make the bacteria susceptible to antibiotics. What if we could take a living organism, so it could be bacteria, it could be a yeast cell, it could even be a mammalian cell, and use this in some way to engineer a kind of a correction, a network, a genetic network, which we could interact with our own bodies and correct some default within our bodies. This is the idea of living therapies. What if we could take out T cells and we could systematically engineer them so that they would attack cancer cells specifically, and therefore you could have a kind of an immunotherapy, and there's a lot of work going on this area. So T-cell engineering for treatments of cancer. What if we could take genetically sort of designed circuits, if you like, and I can tell you a little bit about this later because we're working on this in my lab, and actually implant them into paper strips and use not living cells but cell-free extracts so that we can have a paper sensing device that could detect viruses, bacteria, or parasites. What if we could re-engineer pluripotent stem cells? So this is taking fibroblasts, that's doing proper systematic engineering, looking at those transcription factors which create those fibroblasts into pluripotent stem cells, which then lead into all the different differentiation pathways that we might want. So this is cell therapy and regenerative medicine. What if we could take some of the plant natural products that have direct therapeutic properties, but are difficult to chemically synthesize, so these would be very complex chemical molecules. Let's take all the genes that make them in a plant, but let's take all those genes from whatever source we can, create a metabolic pathway, put it into an E. coli, grow up thousands of liters of E. coli, and produce the chemical through this kind of industrial process. And there's some very good examples of that as well. So welcome to the world of synthetic biology. So those are just some of the examples, I think, which are beginning to emerge from synthetic biology. And I think what is exciting is it has this powerful vision, which Adam mentioned, of merging engineering design practice into the systematic construction of biological systems and cells at the genetic level. So this is a level of engineering which has never been done. So people talk about genetic engineering, but actually there's no engineering in genetic engineering. We can talk about that later. So my proposition is that synthetic biology is engineering biology, and thus the process of systematic design is now allowing the construction of increasingly complex biological design. So the introducing the word design, and that changes the whole way of thinking about how we might manipulate biological systems. And so this is a little bit provocative, but I think the limits of synthetic biology will not necessarily be technical, but societal, in that the applications and utility will need to be proven and accepted. And my question is, do you do that?
Up to you. You can do it if you want. But then you miss your round of applause.
No. Okay, so I'll just, so the last bit was, that is the key bit. In terms of the technology itself, it is an early stage, which Rob mentioned, but it is actually accelerating rapidly. And there's a sort of paradigm shift in how we're going to do biotechnology in the future. And that is happening because of two factors, and I'll finish on this. One factor is the knowledge base that we've accumulated over 40, 50 years of very detailed research and biological research. And the other factor is the ability to synthesize chemically large pieces of DNA at a relatively cost-effective way. So when we can rewrite the DNA program chemically and order it and design it, then we're in a completely different world. And I think this paradigm shift of how we do biotechnology is going to be created by synthetic biology. Thank you.
Thank you, Paul. Okay, so lights up. So there were a lot of what-ifs in there and a lot of good ideas. But the question is one that relates directly to you, because these, as we've mentioned, as everyone mentioned, these are in principle an applied technology. So the question is this: would you be happy to have your genome edited to ensure the genetic well-being of your children? Right? So would you be happy to have your genome edited to ensure the genetic well-being of your children? Those who would be willing to have their genome edited, raise your hands.
Oh, damn.
And let's just do the reverse. Who would not be willing to have their genome edited? So I'm going to go, I think that's about 80-20 panel. Do you agree with those numbers?
70-30?
70-30. That's what... If you say so, 70-30. Okay, thank you very much, Paul. Wait a minute, let me just note that down. Right, and I think you've nicely queued up our final presentation, Susan. There were a lot of what-ifs in there. Take it away.
There were, yes. Thank you very much. So I'm Susan Molyneux-Hodgson. I'm a sociologist of science, and I've been studying synthetic biologists since 2007, which is when we got the first kind of big amounts of money going into the field of synthetic biology. And interestingly, when the initial funding came out for networks in synthetic biology, it was a requirement of the funding bodies that social scientists be involved alongside the scientists and engineers. So this was quite an interesting sort of occurrence. It has happened in some other areas of science, such as areas of nanotechnology, for example. And there's lots of discussion around why that would be the case. Why would you want to include social scientists of various different kinds in scientific work, really quite at an early stage of the process? If I had a lab, I suppose I might ask, why is it that we continue to separate the technical and the social? Why is it that we continue to separate science from ethics? And why is it we continue to separate fact and values? So those are the kinds of questions that we try to explore, and increasingly we're trying to explore those collaboratively with the scientists and engineers. Of course, this debate has beautifully set up the science coming first and the ethics or the social bit at the end. And in a sense, that kind of reproduces something that happens quite a lot, that we expect the science will somehow produce results, and then we can worry about ethics, legal, social stuff at the end. So I'll come back to that in my question in a moment. It's not always clear what becomes a controversial science, and I think the questions we've had so far have already illustrated that. I think my fellow panel members were expecting to be sort of maybe not quite the overwhelming voting patterns that we have seen, and whether this is a phenomenon to do with the polling that we had in the run-up to the elections in May, which came as a bit of a surprise as well. I don't know. Something's gone wrong with the idea of polling, I think. In recent years, we've had the idea of upstreaming of debate. There was quite a famous report came out from Demos some time ago now. But again, this is the idea that publics could have a voice a bit earlier on in the process of doing science. It happens in a slightly patchy way, I have to say. But synthetic biology has upstreamed public voice in quite interesting ways. And so there was a dialogue report published very early on. This was a process that was funded by the Biological Research Council, and it was published in 2010. And it did quite a lot of polling, I suppose. There were also a lot of focus groups. And the set of questions that are bullet pointed at the top of here were the outcomes of the focus groups. If people are interested in reading this, this is a report that's available on the BBSRC website. The people involved in the dialogue were actually mostly interested in the process of synthetic biology as a science. They were less bothered about the products because they weren't here yet. Okay, Paul presented a lot of what ifs, and a lot of synthetic biology is still very much at that stage, what if? And so it's not always easy to reach decisions on what our view might be on those particular technologies. So the focus group showed us that what these particular publics were interested in were how do you make decisions about which areas of science to pursue, who were the winners and losers potentially in these developments, because there are often consequences way beyond our own environments, and how do you know you're right and what alternatives could there be? Okay, so there was actually that's quite an interesting set of questions for publics to come up with, quite sophisticated ideas. But here's another load of questions. So we've got a range of topics of potential application areas here. And my questions really back to you are around, well, how should we go about deciding whether to pursue something or not? So not the debate question of how far should it go, but actually how should we go about deciding how far it should go and in which directions? Who should have some kind of voice in those debates? How should we set up decision-making structures to do that? And I guess it leads to a wider question, not only about synthetic biology, but just what's the relationship we'd like to build in the 21st century between technological developments and us as citizens and kind of involved inevitably in those developments. So I will finish there, and Adam will repeat my question.
Thank you, Susan. Now, Susan presented a lot of questions in there, and I forced her hand beforehand to reduce it into one so we could do this vote. And so the question for Susan is this: should we focus on process or product? So effectively what she was just presenting. So I want people who think we should focus on the process of understanding the science, the scientific process in synthetic biology, please raise their hands now. And I'm assuming the converse is, will people raise their hands if we should focus on the products? Interesting. Interesting. I'm going to go with 70-30 again. 70-30? I need consensus from the panel here. 70-30.
Okay. I'll give you.
Which way round was it? 70 for the...
30-70.
70 products, yes. So it's very, very scientific polling, this. Excellent. So that's all four panelists. Now, I want to do something else before I open it up. I want to say something about, which is my field of expertise, which is the media. My background was genetics, and as a journalist and as a writer, I was drawn towards synthetic biology as a topic worth covering. I did some basic genetic engineering during my PhD 10 years ago, but the field had radically changed over that period in a way that what I was doing in the lab that took me 3 years effectively could probably be done in about 2 weeks by not even an undergraduate, probably by a school-age biologist. Now that shows the reduction in the entry level for the technology that we're talking about now. And a lot of it is to do with the projects that I know Paul is involved with, but I'm presuming that Louise and Robert, you are too, and that's called BioBricks. Maybe, Paul, you'd like to just briefly describe what the BioBricks project is.
I think you're referring to the iGEM competition, which is the International Genetically Engineered Machine Competition, which is a worldwide competition for both undergraduate students, high school students, and overgrad students, an American term for those doing masters or PhDs. And it's been running since 2003, and in the last competition, which was about a week ago or 2 weeks ago in MIT, there was 260 odd teams from all over the world building machines using standardized parts. So what you're referring to in terms of BioBricks is the concept of having standardized genetic parts that you could share you could design with, you could, there are sort of interoperability that when you assemble them, they had sort of standard bits that would stick together if you were kind of, and they have this term called biobricks, so that defines biobricks. So and there's a parts registry. I can't remember the thousands of parts now on there, but it's a very active and very important part of synthetic biology.
So the principle followed on from a lot of the founders of this branch of synthetic biology came from electrical engineering, and they recognized quite early on in the process that instead of having to reinvent every single component in a computer, that by standardizing things like diodes and thermistors and whatever, by standardizing them, they enabled a platform on which anyone could do electrical engineering. And the result, arguably, was the 20th century. So I think the principle in what Paul is just describing is the same. The recognition that every time someone needed to do some or wanted to do some genetic engineering, they didn't have to invent genetic engineering from scratch, which is what I was doing in 2001 during my genetic engineering PhD. So here's a question for everyone, really. That reduction in the entry level, the ability that we have created, that we've built these tools so that effectively anyone can do this kind of thing with a bit of training, has that decreased the conversations that we need to have that Susan was talking about? Has it reduced the need for us to discuss the societal impacts because everyone can do it? Susan, perhaps you could answer that first.
Okay, so has it decreased? In principle, no. There's no reason for any kind of advance to necessarily decrease discussion around some of these issues. In some areas, it's potentially hugely increased it, because we know that there are concerns in some areas around biosecurity, for example. It's perfectly possible to build a microbiology lab in your kitchen or your garage. So this has raised concerns for some sectors, some institutions. iGEM is co-sponsored, or the part of it involves the FBI. They turn up at iGEM every year and do a talk to the students that are there, because they're interested in recruiting the students to take a kind of a responsible approach to what they're doing, and in somehow to kind of safeguard their science from misuse. Okay. So in some respects, there's an increasing need. So if you reduce the entry level, anybody can do it. So yeah, maybe that raises more concerns rather than less.
Louise, I want to ask you what you were talking about in terms of the biodegradation by engineered synthetic organisms. That's a theme that has appeared frequently over the last few years in the iGEM competition. And I mean, it's related to what we were just talking about, but the prospect of creating bioengineered synthetic biology products and then releasing them into the wild is something that I think, well, yours was the most nuanced vote, and we got a 50-50 split. Where are the safeguards in there in terms of the discussions that we need to have before you create a product that we can actually release?
So I'd like to specifically go back to the iGEM competition with regards to that. The iGEM competition is very cleverly designed, that you have to it has different medals are awarded for different stages, and you, as long as you fulfill those criteria, you are awarded that medal. So it brings in straight away the requirement to consider the policy and practices of the application that you're looking at. So what it's actually doing is making the students, the teams, think right from the beginning about that application. And it's the teams that don't do so well that are the ones that haven't thought about it. And the ones that do well, Edinburgh this year did fantastically well, and we actually got the Policy and Practices Prize. Thanks for setting that up for me.
Congratulations.
So, and it's part of the competition that this is the students are taken through a process where they actually learn more about integrating policy and practices than PhD students do, than later stage researchers do when they kind of shut themselves off from and shut themselves in a lab and whatever. And it's actually the field itself needs to look at the way that the competition is structured and considers all those different elements and make sure, and they are doing more and more so, make sure that that is involved in all aspects of training.
Okay, so Paul, you've been heavily involved in iGEM since its inception,
Pretty
since
much,
2003
or
three or four.
yeah.
Yeah, it varies. And in fact, it was held at, the big conference was held at Imperial in your lab, effectively, 2 years ago. 2 years ago?
Yes, SB 6.0, yeah.
Yeah, so we're getting to the stage now where the people the undergraduates who are involved in the iGEM project competition, almost 10 years ago now, are getting to middle stage careers in science. So the question is this, and I want to ask both you and Robert this. That framework that Louise was just discussing, where the ethical considerations and the societal considerations were inbuilt into the competition itself and rewarded, are we seeing those effectively in now research scientists who are still involved in synthetic biology? It feels like a bit of a loaded question. So
No, it's not. I'll just be straight up.
And Susan.
The question is, yes, it is. I think the thing about iGEM is that there's about 20,000 alumni, if you like, around the world. So there are 20,000 students who've gone through that competition. And some of them, as you rightly say, some of them are now, assistant professors or lecturers or university appointments. Some are in research in industry, some are writing book, whatever. But they've had that experience, and it's absolutely integral to synthetic biology, that experience, because a lot of the human practice stuff which Louise talked about is really a major part of the synthetic biology field. And I can see that happening. So when we have social science collaborations, we have debates. We've just been to one all day actually, where our PhD students were talking. We're exposed to this discourse between professional social scientists, really challenging. What are you doing? Have you thought about all the knockout? All this sort of stuff. So it's a really healthy part of the field. So the quick answer is yes. I can see this as a very major impact on how we're going to do this field.
Robert, a slightly different question to you. Is this new? Right? Is this what we're talking about now? Susan talks about how she's studied synthetic biologists now for a few years. Is this new? Is this the first time we've really done this type of endeavour where from the get-go we've tried to engage the public successfully or otherwise?
I think what synthetic biology has helped to do is raise the awareness of the public being interested in what people are doing in the lab. And I certainly see a complete difference in the youngsters coming into the subject and their view of how what they're doing relates to society in a way that is completely different from it was 10 years ago. In fact, one of the things that came out when we did the synthetic biology dialogue was some postdocs became quite incensed by the idea that people might want to interfere in what they were doing. Members of the public might have in some way interfere with it. I think that's a way of thinking. I think it's changed a great deal. Certainly in my experience working at Newcastle and also at York, people are much more tuned into public opinion.
I want to ask the audience this. So in many ways, I represent the media here, and I'm not neutral on this subject, and there is a great deal of self-interest. I did write a book about synthetic biology, which is actually called Creation, not Creativity, but it doesn't matter. And in fact, I made a Horizon in 2012 about, this is relevant, I'm not just this is
on
relevant. I'm not just plugging my own stuff. But I'm not sure what the level of, how you feel the level of coverage of synthetic biology has been. We've been doing this for 10 years or so. So I've got a question for you, and I want a quick vote. So do you feel satisfied by the level of coverage of synthetic biology in the way that it's informed the whole conversation that we're having now? I realize it's a bit skewed because we're doing it now. It's a bit meta, this. But if you could raise your hand if you feel satisfied by the media coverage of SynBio over the last, 5 years, 10 years. Hands? No one? Well, I've got to say, that's a little bit disappointing to me. Nobody. Come on, someone in the
dear.
gods. That doesn't include you, Martin. Right.
Never mind.
Did anyone see the horizon? One person. 4 people. Dear Lord. Has anyone bought my book? No, don't answer that. Will anyone buy my book? Oh, come on! He's in it. Right. I'm going home now. On that note, now there's one more question I want to ask before I open it up. So I was going to say there's been a lot of hype about this field, but clearly there hasn't. It is a subject that has been discussed a lot within scientific circles. It has. There have been accusations clearly amongst scientific circles. Seriously, I'm really disappointed by that. It was repeated about a month ago. It's still on the iPlayer. No one. Where are the applications? So I framed this as being a discussion, a field which is defined by being applied. We don't necessarily all agree about that, but with engineering at its heart, the applications are really crucial to the success of the synthetic biology project. Now, does anyone want to give me an example of a synthetic biology product which exists in the real world?
We could introduce the artemisinin.
Shall we introduce artemisinin?
But, I mean, it's so fraught with huge debate, and I know my colleagues will debate. So artemisinin is an anti-malarial drug. The Nobel Prize was awarded 2 days ago or 3 days ago for a Chinese scientist who, unfortunately, I can't remember the name, but she discovered the active ingredient. It's an anti-malarial treatment. It's grown in wormwood plants, I think.
2UU.
2U, thank you, in wormwood plants, I think, and it's naturally extracted. So an American scientist called Jay Kiesling has been working maybe for many, many years trying to work out if you could, like the slide I showed, take the metabolic pathway that produces artemisinin, but instead of growing these plants and harvesting them and extracting them, and let's manufacture it in an E. coli or a yeast cell, and let's grow big vats and have kind of production of this pharmaceutical. And that's now been done, and One World Health and Gates Foundation are involved, and there is millions of tablets of artemisinin now being produced using that synthetic biology engineered yeast organism.
Yes, yes. Although that's not quite right, though, is it? Because they may be being produced, but so that was first done. The first paper.
This is why I said it introduces a huge amount of debate.
Well, it was first published. That pathway was first. It's a good story. This. If only someone had written a whole chapter in their book about it. It's a good story, this, because the first paper that described the synthetic pathway was 2009 in yeast. And immediately, people like me and the press went, right, well, that means we're close. So more people have died as a result of malaria than any other single cause in the history of humankind. Right? And the prospect was, as you mentioned, the Chinese... Not weed? No.
Wormwood tree.
Wormwood tree. There you go. Yeah. So it's a slow-growing crop. It's very difficult to harvest, and you get low yields. The prospect was, from Jay Kiesling's company, Amaris, based in San Francisco, was that we could reduce the cost of individual treatments for malaria via artemisinin, synthetic artemisinin, by tenfold to 25 cents. Right? So everyone wants that, of course. And that's why the Bill Gates Foundation got involved and dedicated 40-odd million to it. Now, that was in 2010. And I don't know whether you guys know the joke about nuclear fusion has been a technology which has been 30 years away for the last 60 years or so. Well, it feels the same to me. We're 6 years on from artemisinin's first description as a synthetic pathway.
It's been manufactured in Italy. There are huge industrial, centers that are producing this product. I've seen them. I've seen the packets. I've seen the tablets. There are millions of them being produced. And Jay gave a talk in Singapore about 3 weeks ago about this. It's full of controversy for all sorts of reasons because, we can go through the debate. The farmers are going to have to replant, displaced farmers. Local indigenous populations grew these trees that they could harvest the active ingredient and all of this stuff. You're probably best placed to critique this because a lot of the social scientists have critiqued this quite significantly, actually.
Yes.
Whereas actually, it's a fantastic project by a really excellent scientist, actually.
In my opinion. We can talk about the farming argument, which I don't think, well, I think is specious, but I think the more significant argument is that artemisinin resistance has already emerged in several locations, and that is not a scientific problem, that's a distribution problem. Anyone want to comment on that? No. Right. I'm glad we got that sorted. It's also worth noting that it was a serendipitous discovery that actually the company was primarily concerned with producing artificial synthetic diesel, and a similar pathway was involved, and a postdoc in his lab noticed that one of the byproducts would be a precursor to artemisinin. That never came to fruition. Did it?
No, they did. They produced. Yeah, they did. Yeah, they're in Brazil, and they're producing farnesene, and they're producing biodiesel, which is actually, I think, there's 20,000 buses in wherever, São Paulo, that's been powered by that molecule. No, they've got a big plant down there.
I was trying to introduce debate.
I don't know why I know.
So I think one of the things we could maybe ask here is, we can do things, science can do things with synthetic biology. How successful these applications are at the moment is open to debate. And so is that about we were just doing something because we could, and there weren't the markets, or there weren't a kind of a set of circumstances into which these applications could be launched? I think the hype has very much been within the scientific community and with policy communities as well. There's been a lot of investment in synthetic biology. It's expensive to do, and as with most science, you have to compete for resources. And so we hype things. This is obviously the royal we. I'm not a scientist. That we need to draw resources to our work, and so we make promises. There are undoubtedly lots of societal challenges that synthetic biology could address, but again, we've been here before. The earlier versions of biotechnology promised to solve a lot of problems. Nanotechnology promised to solve a lot of problems, and now synthetic biology is promising to solve a lot of problems.
Well, where do those promises come from, though? Is it people like me?
Some of it will be you. Yeah, I watch your programme.
The only bloody one in the whole room.
I particularly like the spider goat. But I think, a lot of it is the technical communities as well, because you have to get resources to do your work, and so increasingly there's less money around. And so if we want to kind of make a change in the world, we have to find a way of getting the money to do the work. So of course we're going to promise stuff. I have to promise to have an impact on stuff as well.
Well, you are now. Oh, Robert.
I think it's quite interesting that two of the cases that we've been talking about where the fantastic technology, it works, but they're disruptive technologies. They're moving into spaces which are occupied by alternative routes to production. Artemisia can be produced quite nicely from plants, and there's been a big breeding program to increase artemisinin content, and you can very easily produce diesel from a variety of different chemical and biological processes which don't require synthetic biology. I'm not sure that we've chosen the best examples to go forward to show what it can deliver. I think we've tended to go with things, low-hanging fruit that look like a good idea at the time, but perhaps don't ring too many bells when they do come onto the market.
Do you include artemisinin in that?
Well, it's a success. I mean,
it's technically extraordinarily challenging. And I've seen these great big vats producing this chemical, and this photoactivatable last step where it changes the structure, and it's secrete. It's a brilliant example of what synthetic biology can do. Now, the question is more about, was it the right solution was there a problem that it served? And I think there are arguments on the other side that say there was, because the production, and you can argue about this, but the production processes were very up and down. If you look at the production of artemisinin over a 10-year period, it was all over the place, depending on the crop.
It was a classic boom and bust cycle.
And so it was stable. I mean, we shouldn't really spend all the time talking about artemisinin, because there are great projects like biosensors that are being done at the moment, particularly the arsenic water biosensor. But I think you're missing the point. I think it's a paradigm shift in how we're going to do biotechnology. That's the point. There are whole bunches of companies happening in California now that have huge amount of investment into automate all the steps that you did in your PhD that took you 5 years. We're not going to need, postdocs going like, really intelligent, highly educated people going and taking little tubes, putting them in a microfuse, going around a centrifuge, and putting it. We're not going to need those kind of people anymore. We want people to think about design, not this idea of pipetting liquids and measuring pHs. And so what's happening, and I think this is the reality, is there is a shift to get rid of all that. We're going to automate all of molecular biology pretty much, and it's going to be driven by this design process, and that's happening. So that actually is happening, and I've seen that as well. So if we just, go back in ourselves and say, well, actually, it's a technology that promises, and the UK kind of sits there thinking, well, are we going to be part of this? And you just need to go to California. I mean, this debate does not happen. They know where they're going. They're going to create a whole new industry.
But I wonder whether that is a cultural difference that is born of the history of this field.
Yeah,
so
maybe.
there was a great debate. Well, it wasn't a public debate, but before, right in '75. There was a specific meeting to address the societal concerns of effectively all of the genetic engineers on Earth, which was about 100 people at that time. And they invited lawyers and they invited journalists into this room in California, the Asilomar conference. And much of the analysis of the result of that was that these types of conversations in America don't happen. There was great support for this neonate technology. Now, we never did that in this country. And I wonder whether there is a fundamental difference in our attitude towards genetic engineering and, as a result, synthetic biology here in Europe, as opposed to the States. That's a question for anyone.
Yeah, well, they're completely different cultures. Of course they're going to be different.
But they're not completely different cultures. I mean, that's a slightly peculiar thing to say.
They speak a version of English,
yeah. Well, explain yourself. Why are they completely different cultures that would react in such a different way?
I don't really know where to begin to answer that. They have fall and we have autumn. Except, of course, fall was what we used to call it in Shakespearean times. They just kept the word, and we changed. So scientific cultures vary hugely, even between labs. I could go into Paul's lab, Robert's lab, the labs that I go into, and they operate in noticeably different ways. So the cultures of California are going to be massively different to the cultures of Imperial and Edinburgh and Newcastle. That people are trained differently.
There were Cambridge scientists present at Asilomar, though.
Yes, I'm sure they were, and in many respects, you were saying how the stuff that you did as a PhD student has changed rapidly, and that was in 2001, and now you're expecting 1975 to be somehow a relevant reference point to what's going on now. The world's changed a lot since 1975.
Undoubtedly, but are you saying it's not relevant? I recognise that the major difference is that the entire community of genetic engineers in '75 was about 100 people, actually a bit more than that, and now it's the population of a medium-sized city. I understand that those two things are different, but this is effectively what we're trying to do in conversations like this, though, aren't we? We're trying to engage scientists with society. So it's...
Asilomar didn't invite... it wasn't an open invite to buy a ticket and come and debate. It was a fairly enclosed kind of set of discussions, which did kind of come out with the idea that the scientific community could take responsibility for monitoring itself in relation to the technologies it was developing, whereas now we're in a much more open forum.
Well, the exact same model was adopted 3 years ago when the H5N1 when the experiments were done in those two labs in Holland and somewhere in the States. to shut down, to have a moratorium on the research in order that they would model exactly what had happened in Asilomar in 75, such that they would learn from the successes of that project.
I wonder, is there any feeling within the scientists on the stage whether or not that bioethics is getting in the way?